Good day. Today our lecture about viruses, causative agent of acute respiratory viral infection. Acute respiratory viral infection are acute viral disease characterized by predominant lesions of upper respiratory tracts. The causative for widespreading airborne contamination, a great variety of causative agents, absence of immunity to recurrence contamination. Taxonomy of viruses. Taxonomy main viruses which can cause acute respiratory viral disease. Family Adenoviridae, genus Mastadenovirus and genus Adenovirus, type of nucleic acid DNA, organization of viruses, simple viruses or naked viruses, type of symmetry, cubical. Of family Orthomyxaviridae, genus Influenza virus, type of nucleic acid RNA, chemical type of symmetry. Paramyxoviridae family includes two genus Pneumovirus and Morbillivirus. It's RNA viruses with chemical type of symmetry. Picornaviridae includes two groups of genus Rhinovirus, causative agent of acute respiratory viral infection, and causative agent of intestine disease, enterovirus. This RNA virus with cubical type of symmetry. And we know about Coronaviridae family. Genus Coronaviruses, these RNA viruses with chemical type of symmetry of nucleic acid. The main causative agent of an acute respiratory disease: lesions of the nasopharynx, lesion of the stomatopharynx, acute process in the larynx and trachea, more frequently caused by parainfluenza virus, bronchitis, and pneumonia. The common features of acute respiratory viral infection or acute respiratory disease: widely spread in nature, virus low resistant to the environment, are highly contagious, viruses resistant to the environment are low contagious. Route of transmission airborne, except adenoviruses and coronavirus, which may spread by a fecal-oral, out or via contact. Main common features: clinical manifestation are similar: lung inflammation, inflammation of the respiratory tract, and symptoms of intoxication: weakness, headache, fever, and other symptoms of intoxication. Epidemiology: a source of infection, sick individual or sick person. The main mechanism of infection transmission aerogenic, frequently via contact. For some viruses, adenoviruses, coronaviruses, in fact, may spread via local oral contact. Routes of transmission airborne, during coughing, sneezing, less frequently via an air-dust-borne way. It has also been proved that some causative agent of acute respiratory viral infection may be transmitted via contact: adeno-rhino respiratory syncytial virus. Virus and disease: adenovirus cause affect the nasopharynx, alveoli, intestinal mucosa, can cause pharyngoconjunctivitis in children, and can cause enteritis. Parainfluenza viruses cause affect the larynx and hoarse voice, laryngitis. Rhinoviruses cause effect of nasal mucosa, contagiosis, rhinitis. And influenza viruses attach of mucous cells of the trachea and bronchi are affected. Immunity. Localized production of interferon, the factors of non-specific immunity. Virus neutralizing specific immunoglobulin A provide local specific immunity. Cellular immunity. Antibodies are usually generated too slowly, which makes them low efficient in the struggles with an acute process. In essential features of acute respiratory viral infection is formation of secondary immunodeficiency. In most types of acute respiratory viral infection, post-infectional immunity is unstable, short-lasting, and type-specific, with the exception of an adenoviral infection, which is accompanied by the formation of a fairly stable but also type-specific immunity. For specific prevention of disease, viral, bacterial disease, we used vaccine. We know different type of vaccine: live attenuated vaccine, inactivated vaccine. Live attenuated vaccine can be recombinant peptide vaccine, recombinant live vectors vaccine, conjugate vaccines, virus-like particles vaccine, and another type of vaccine which you use for specific prevention of different disease, viral infection or bacterial infection. And now we will study disease and causative agent of disease. Influenza is acute polyinfection with an airborne ways of transmission. It's characterized by a lesion of mucous membrane of the upper respiratory tract and by severe intoxication. Its prevalence may be epidemic or pandemic. I will not read about a historical reference, but you have to know we know 3 pandemic of human influenza viruses in 1918, 1957 and 1968, and one global epidemic 1977. Taxonomy. Family Orthomyxoviridae. From Greek ortho, correct, myxa, mucosa. Genus influenza viruses, and includes type A, B, and C influenza viruses. Subtype A, type virus of influenza by hemagglutinin, includes certain subtype age from 1 to 30, and by neuraminidase classified up by 10 subtype. Neuraminidase, later M, from 1 to 10. Types of influenza viruses we talk about type A, type B, and type C of influenza viruses. Different basic features of these viruses, this type of influenza viruses: different symptoms, age group, effect, divided, epidemics, and experienced. Epidemic, my student, type A virus of influenza viruses very often widespread and can cause pandemic. Type B, regional spreading of influenza, and type C don't cause. Genome: single-stranded minus RNA twisted into a double spiral with single spiraled edges. Consists of 8 fragments. 5 fragments encode synthesis of 1 proteins, and 3 fragments of 2 proteins, totally 11. Type of nucleocapsid symmetry, helical or spiral. Morphology: spherical, oval, less frequently filamentous and spermatozoidal forms of influenza viruses. Dimensions: diameter of A viruses of influenza is 18 to 100 nanometers. Of B and C viruses from 100 and 200 nanometers. Complex structure: it consists of a capsid covered with envelope. On this slide you can see fragment type of RNA. Around RNA you can see capsid, and around capsid you can see envelope. On surface envelope we see glycoproteins, hemagglutinin, and neuraminidase. Flu viruses are named by the type of surface proteins: hemagglutinin, trimer hemagglutinin. Herpes viruses enter cells. Type A infect humans, birds, and pigs. Type A has more than 20 different subtypes. And neuraminidase, hemagglutinin, you see, neuraminidase, you see, tetramers, and help viruses exit cells and classified by 9 subtypes. Flu viruses, common full name of human viruses: human 1, H1N1, H1 N2, and H3N2. Influenza viruses contain protein M, matrix protein, encircle the nucleocapsid, bind in the supercapsid. On envelope there are spine about 900 of 2 types. I told hemagglutinin recognize sensory cell and attach of their surface, and neuraminidase, splitic sialic neuraminic acid, provides the penetration of the viruses into the host cell and living virions. Nucleocapsid, RNA and capsidal proteins. NP proteins, ribonucleoprotein, differentiates the viruses, influenza viruses, into type A, B, and C. Internal proteins, PB1, PB2, and PA, are proteins of a polymerase complex. They participate in transcription and translation of influenza viruses. And on this slide you can see the scheme of influenza viruses, segmented RNA. Around RNA you can see capsid, and around capsid you see envelope which contain hemagglutinin, which help of viruses attach of surface sensor cells, and neuraminidase, this invasive function of viruses. On this slide you can see more information about a structure of influenza viruses. Chemical compositions of the viruses: 17% of proteins, 20% of lipids, 5 to 8 of carbohydrates, and 1 to 2 of RNA. Antigen structure of human influenza viruses: S antigens, V antigens, and hemagglutinin and neuraminidase spikes. Variability of influenza viruses. Why? Viruses have different subtypes. Variability is inherent in envelope antigens. Two genetic mechanisms are distinguished: antigen drift, insignificant change that are caused by point mutation, more frequently in hemagglutinin. This change leads to the development of distinctions in strains. As a result of this process, epidemics occur, frequency of occurrence each 1 to 3 years. And a second mechanism of variability genetic material of influenza viruses, this shift, is an absolute replacement of a gene. Generation of a new antigenic variant as a result of a genetic recombination between the viruses of humans and animals. This leads to substitution of type hemagglutinin or neuraminidase, which cause pandemics. Frequency of occurrence is 10 to 20 years. On this slide you can see how viruses can change genetic material. For example, human viruses ARD2 and 2. After genetic variability, antigenic shift, genetic structure of human viruses interact with genomes pigs viruses, animals viruses, and forming new subtype of viruses, human H3 and 2. Every H3 and 8 viruses point mutation of hemagglutinin and neuraminidase genes during antigen shift change cause point mutation. Viruses B and C of influenza are devoid of the shift type variability. Viruses B of influenza cause epidemics. Viruses C of influenza cause sporadic disease conditions or local outbreaks. Specific features of the virus's reproduction. We know stage reproduction of viruses and first stage adsorption on the receptors of the sensory cells contain sialic acid with the help of hemagglutinins. Penetration into a cell via endocytosis. Deproteinization gets free from the encapsidal proteins. An eclipse phase, replication of nucleic acid in the nucleus and synthesis of virus proteins in the cytoplasm on ribosomes. Assemblage of the nucleocapsid in the cell's cytoplasm. Exit from the cell via budding or lysis. Air envelope is formed from the cytoplasmic cell membrane. We know about it. On this slide you can see stage of reproduction or stage of multiplication viruses in host cells. You see different these human influenza viruses, stage of attachment and penetration. Third steps, this RNA or ribonucleic acid travels to the nucleus where is replicate user force, and another steps viral messenger ribonucleic acid after that forming new viral RNA, and next forming neuraminidase antigens and hemagglutinin and release from host cells. Cultivation of viruses, method of laboratory diagnosis. Cultivation in the bodies of sensible animals. We use white mice, rats, ferrets, and other animals in chicken embryos, and we will use for cultivation of viruses, influenza viruses, in a primary culture of embryonal kidney cells of humans, some animals, and in the twisted cells. Resistance of influenza viruses. Stability to the environment is not high at the room temperature for several hours. At 56 degrees for 5 to 10 minutes. At 100 degrees, immediate death. Sensitive to drying out procedure, ultraviolet and disinfectants are well preserved in deep freezing. Epidemiology. A viruses of influenza cause disease in humans, mammals, and birds. B and C viruses of influenza cause disease only in a human. Source of infection: sick individuals. Mechanism of infection or route of transmission: aerogenic. Less frequently by contact. Ways: indirect contact. Seasonality of disease: autumn, winter, spring period. Incubation period of disease from 1 to 2 days. Pathogenic and clinical features. The mucous membranes of the upper respiratory tract cause selective lesion of the cells of the cylindrical epithelium. The symptoms of rhinopharyngolaryngotracheitis: dry cough, sneezing, rhinitis. After the destroy of the affected cells, disseminated into the blood canals, cause viremia and clinical symptoms: fever and intoxication. Suppresses the immune and hematopoietic system, cause secondary infection or anemia. On this slide you can see different symptoms: fever, AIDS, chills, and other clinical symptoms. And what cells can attach viruses: nasopharynx cells, respiratory cells, gastric cells, joint cells, muscular cells. Systemics cause fever and weakness, and central nervous system cause headache. Immunity of post-infection immunity is humoral and cellular, tense, long-standing, but type and strain specific. Microbiological diagnosis: test material washing and swabs per nasal, pharyngeal, conjunctival, sectional material, and the test material become infit. For express diagnosis, rapid method of diagnosis of influenza viruses, we use reaction of immunofluorescence and ELISA or test or immune enzymes analysis. Virological technique, we can use chicken embryos, cell culture, and contamination of test animals. Indication in tick embryos: reaction gametabsorption with thickened erythrocytes. In cell culture, we will see cytopathic effect. In the test animals, we see clinical features of infectious disease, which cause death of laboratory animals. For identification viruses, we used CFR, complement fixation reaction, and reaction chemagglutination inhibition. Next method of study viruses: serological technique, determination of antibody titer in reaction chemagglutination inhibition, complement fixation reaction, and reaction immunofluorescence. A molecular biological technique, we used PCR. For specific prevention of influenza viruses, we use live inactivated whole virions, inactivated split vaccine, and inactivated subunit vaccine. We see different name of vaccine and different administration route of this vaccine. On this slide you can see different vaccine, type of viruses which contain of vaccine, routes and doses. For specific treatment of influenza viruses, we can use antiviral drugs with two main mechanisms of action: inhibition of viral uncoating and penetration, and neuraminidase inhibition. This amantadine, rimantadine, it's antiviral drugs with inhibition of viral uncoating. Oseltamivir and zanamivir, neuraminidase inhibition. In several cases of anti-influenza donors, immunoglobulin is used for passive immunization. And next virus is para-influenza viruses. It's an acute infectious disease characterized primarily by lesions of the upper respiratory tract, by fever and moderately mild intoxication. Taxonomy: family Paramyxoviridae. Genus Paramyxovirus and Rubella-virus. Type para-influenza viruses first and third, and rubella viruses include second and fourth type. Genome strategy: non-segmented, single-stranded linear minus RNA. Type of nucleocapsid symmetry: spiral. You see, for example, this nucleic acid. Around nucleic acid you see capsid, and around nucleocapsid we see envelope. Morphology: complex structure. Complex structure is envelope viruses. Surface envelope, glycoproteins, neuraminidase, hemagglutinin. Activity of hemagglutinin and neuraminidase, adhesion function. Parainfluenza viruses contain F proteins or proteins of fusion. Fusion of the virus membrane and the sensory cells membrane. Symplasta forming, hemolytic and cytotoxic activity. The nucleocapsid is enriched by M proteins, RNA plus nucleoproteins, polymerase proteins P and L. Chemical composition: proteins, lipid carbohydrates, and RNA. Antigen structure: the internal S antigens, the external V antigen. Specific features of the virus reproduction: adhesion of sensory cells due to neuraminidase hemagglutinin, and multiplication of viruses in the cytoplasm of the cells. Cultivation: developed purely in chick embryos, cultivated in primary trypsinizated renal cells of monkey, human embryo, and another cells. Resistance viruses at the room survives for 5 to 7 days, sensitive to infertile environmental factors. Epidemiology: source of infection: sick individuals and virus carriers. Mechanism of infection: like another viruses, aerogenic, less frequently via contact. Seasonality: autumn, summer. Incubation period of infectional disease from 2 to 7 days. And about type of influenza viruses. Parainfluenza viruses very often cause laryngotracheitis in infants and young children. Parainfluenza viruses of slightly less significance than parainfluenza 1 or 3. It has been associated with group primary in children with mild upper respiratory infection and associationality with acute lower respiratory disease. Parainfluenza third is a minor cause of several lower respiratory disease in infant and young children. And parainfluenza 4 is the least common of the group. It is generally associated with mild upper respiratory illness only. Pathogenesis and clinical features. The viruses take root in the cells of mucous membrane in upper respiratory tracts, destroy them, and causing an inflammation and edema of the larynx, cough, hoarse voice. Virusemilia stage, intoxication, fever, clinical symptoms in children, particularly under 1 year of age. Complication occur bronchopneumonia and false group. Immunity post-infectional humoral, type-specific, lasts for several years. Specific prevention and treatment have been not worked out yet. Microbiological diagnosis, test material, washing and swab from the nasal and laryngeal mucosa blood. Expression technique, reaction immunofluorescence, virological technique for cultivation and isolation and identification of viruses, serological technique we use for study antibodies, and molecular genetic technique we use PCR technique for study genetic material of parainfluenza viruses. And next viruses in our lecture, respiratory syncytial virus, RSV. Respiratory syncytial viruses is classified as pneumovirus within the paramyxovirus family. Its name is derived from the ability to produce cell fusions in tissue culture, syncytium formation. Unlike influenza or parainfluenza viruses, it possesses no hemagglutinin or neuraminidase. The viral structure is similar to parainfluenza virus, except that envelope glycoproteins are an attachment G protein and the fusion F protein. The RNA genome is linear, non-segmented, negative sense, and single-stranded. Among there is two matrix protein on the viral envelope. Respiratory syncytial virus, like other paramyxoviruses, replicates in the cytoplasm and cause syncytium formation of the cell culture. On this slide you can see structure of respiratory syncytial viruses. The antigens of the surface spike of the viral envelope includes, I talk about its G glycoproteins, which mediate virus's attachment to the host cells, and the fusion F glycoproteins, which induced fusions of the viral envelope with the host cell surface to facilitate entry. F glycoprotein is also responsible for fusion of infected cells in cell cultures, leading to the appearance of multinucleated gigant cell syncytium formation. Antibodies directed at the F glycoproteins are more efficient than anti-G glycoproteins antibodies in neutralizing the virus in vitro. Two glycoproteins mediate attachment and syncytium formation. On this slide you can see what does it mean, syncytium formation, gigant cell, and into gigant cell you can see a lot of nucleus. This reaction of immunofluorescence, you see viruses, light green color, and this cell of human. Stage of life cycle of influenza viruses on this slide you can see. And next slide, list to antigenic subgroup A and B of respiratory syncytial virus are known to exist. This dermatrophism is due primarily to difference in the G glycoproteins. Respiratory syncytial virus is the single most important etiological agent in respiratory disease of infancy, and it is the major causes of bronchiolitis and pneumonia among infants under 1 year of age. Clinical aspect I will not read because you will study during our laboratory class. I will ask you about it. And method of diagnosis infectious disease, rapid diagnosis of respiratory viral infection can be made by immunofluorescence or immune enzyme detection of viral antigen, or by PCR for detection genetic material of respiratory syncytial viruses. The virus can also be isolated from the respiratory tract by prompt inoculation of specimen into cell cultures. Syncytial cytopathic effects develop over 2 to 7 days. Severe diagnosis may also be used but requires acute and convalescent sera and is less sensitive than antigen detection method, PCR or cultural. Treatment and prevention supportive treatments is indicated. No vaccine, monoclonal antibody and immunoglobulin used for prophylaxis. Non-specific prevention of acute respiratory viral disease, you can see wash hand, protect on mask, cut nails short, warm water, and another method of non-specific prevention which cause spreading viruses from human population. Thank you for your attention. Don't forget, take the computer test in your personal account.